So my name is Hai Deng, and I'm actually from Vietnam. Growing up in Vietnam, I moved to India for school. So I went to Woodstock International School in India for 6 years. So I live in India there. That's where I actually was really much developed into kind of very interested with math and chemistry. And do a lot of extracurriculum, kind of like high school sort of thing. High school people, how many people like math? Raise a hand. Okay. Chemistry? No? Biology? Okay, good. Biology. Anything other science related? Okay. Engineering sort of thing? Physics? Yeah. Okay. Cool. I kind of like physics too. I was very focused kind of in high school to kind of really go into science. And what really got me interested in kind of a dream job was in biomedical science was actually nothing to do with classes, but it was actually because of the jazz band. Because one year we actually went down to New Delhi in high school and in India, and we actually performed jazz band for a missionary hospital where it's a leprosy mission. And so a lot of patients there that are coming in, they actually have been outcasted from the society just because of leprosy. And so that what really got me interested in kind of trying to figure out why do patients actually get leprosy in the first place, and how can we actually prevent it from people being outcasted from society? And so with that in mind, then I kind of moving on to college. I moved to Ohio, to College of Worcester, which is a small liberal arts college. There I major, you see, like BA instead of BS, in Bachelor of Science in biochemistry and molecular biology. Kind of a random thing, but actually minor in dance and theater, kind of keeping up with my performing arts. So favorite courses, of course, dance and biochemistry, but kind of like what you kind of very potentially find very surprising for me was the barrier that I need to overcome was actually passing biology and biochemistry. It's just one thing was biology was not my main focus. I was very much hardcore with biophysics, biochemistry, but I just realized that if I wanted to be in biomedical sciences, I'd have to challenge myself to actually learn biology. And it was really out there for me too. So in a way, just have to keep plugging in it. I'm still doing it today, even though it's like 10, 15 years later. So from my undergrad, I actually moved here to University of Minnesota in 2006, where I actually did my Ph.D. in biochemistry, molecular biology, and biophysics, so BMBB department here at the university. And then once I graduated here in 2012, I moved to Boston, where I was a postdoc at Harvard Medical School. And I was employed in Massachusetts General Hospital, or MGH, within the Cancer Center. And how I got really interested in that was really trying to understand how do we use the biochemical processes and things that we are learning in cell signaling, and how do we apply that into cancer biology. And as I just alluded with you, I actually just moved here last week and started as an assistant professor here in the Department of Pharmacology. Literally started Monday, so this is day 5 on my job. So the problem in my lab that I wanted to solve is I have been focusing on this problem of DNA damage response and understanding how cells respond to different types of DNA damage. I'll give you a little broad overview about that a little bit later in the next slide. But really understanding what are the signaling mechanisms in cancer that we can actually learn about the mechanisms in order to find new targets to develop new drugs and new ways to target those cancer cells specifically. You have heard a little bit earlier about mutations in different cancer types. So a lot of the mutations out there in different cancer types are also mutations in DNA damage response pathways. So why are some mutations so prominent in breast cancer, but then why are some other mutations are actually in pancreatic or prostate cancer, but they give rise to completely different cancer types? So how do we need to target individual patients as well as the cancer subtypes within that? So that's kind of the focus of my research gonna be. Hobbies, I actually still do a lot of dancing and choreographing, so I perform a lot in Boston as well as here in Minnesota. So here's kind of a big, brief overview. Did you know that every single cell in your body have to deal with about over 100,000 DNA damage lesions per day? Every single cell in your body has to do that. And so you are being exposed to different type of environmental radiation. There's normal radiations going on. UV damage, if you go outside in the sun, you will get some kind of DNA damage. And that's how your cells have to deal with every single day. So wear sunscreen. So that's one thing I would tell you too. So in a way, your body have all of this DNA damage that can occur. And so the cells have to figure out ways to deal with these problems. And because there are so many different types of DNA damage that can occur per cell, there has to be a way that different mechanisms is in place to repair these different processes. So there, what we call in the DNA damage response field, or DDR field, is that there has to be a way to sense them, and there has to be a way to coordinate the different type of repairs that are going on in order to actually making sure that your genome is intact without any kind of mutations. However, if the cells have so much DNA damage, they can actually induce cell death. So if there's so much problems, you want to actually kill off the cells so that it doesn't give a problem. However, that could be a problem too, because there are neurological diseases that actually have mutations in DNA repair genes that are associated with this problem because cells are dying, which is not really good for that problems. However, in the cancer and aging aspect, you actually accumulate of these mutations in these cells. And so how these arise is that the mutation in genes that are actually try to repair, either try to repair the problems, the DNA damage, or mutating in genes that are actually stopping them from actually killing those cells. So then you actually have then cells that are then now can proliferate and grow, and then actually can causing problems, and that's how cancer can actually progress. So in the lab, what we're really focusing on is multiple things. Number one is what are the sources of DNA damage that can actually occur by the different types of DNA damaging agents. And by the way, if you look at cancer therapy, a lot of the chemotherapeutic agents are actually DNA damaging agents. So we also need to understand how cancer cells will respond to these different types of DNA damaging agents, and what are the vulnerability and what are the proteins and genes that are involved that we can actually target to actually selectively killing them. So the first things we wanted to do is understand what types of DNA damage there are. And the second aspect in the lab is really understand what are all the different mechanisms that are there to actually help to prevent or repair this type of DNA damage. Particularly in cancer cells, if they are very important for cancer cells to repair these lesions, and what are the pathways for that, we can then actually target those pathways to actually selectively killing those cancers. So that's kind of the broad overview of my talk. We actually develop we're planning to develop a lot of high throughput. I don't know why I'm saying we, because I don't actually have a student in my lab. It's only me right now. So, but the lab will be generally trying to develop high throughput mechanism to do genome-wide genetic screens on different cancer types with different particular mutation per cancer types, to actually trying to see whether we can develop biomarker that can predict in patients where the cancers are and whether we actually have a way to do targeted therapy. So we do drug screening sort of thing, as well as genetic screenings. And with some of you may have heard is that if we do all of this genome-wide sort of studies, we actually need to have computational biologists, because now we have so much data out there that we need to have, programs and software and engineers to actually figure out how we can actually go with this whole analysis. Things you might have heard about is artificial intelligence or AI, that now we can actually just do it by eye, but we actually need machine learning to really picking up all of this minute details that we can go through from different images, that to pick up whether there are subtle changes between cancer cell versus normal cells. And so that's kind of the whole field in terms of trying to figure out developing new therapeutic strategies for personalized medicine. With that, I can take any questions. Oh, yeah. Sure. How hard do you have to work to get a postdoc at Harvard? Pretty hard. A lot of it, I think, in terms of postdocs, or in terms of like graduate school in science, is that you really learn about logical thinking. And one thing that I would really encourage you is actually always ask questions. I'm still learning. There's a lot of colleagues here that I'm still learning a lot from, biologists that I'm not very good with, or even physicians. And so things that like that is not about like me, myself, or you alone anymore. It's about how do you interact with people. You know, it's like on a job. You work in a team. So how do you work within a team? What do you bring to the table? What do you contribute to it? And how do you think about the problems? And it's a very logical way of trying to address problems. So always be curious, always ask questions. I'm still like the stupid one in the group all the time. I'm like, I have no idea what this is all about, but you know, that's my questions because I wanted to learn about it.